each myofibril has alternate dark and light bands on it. Just what we have discussed. The skeletal muscles are striated. They have striation. So definitely the myofibrils need to have alternate dark and light bands on it. So we are discussing about the ultrastructure of skeletal muscles. What we find is that the light bands contain actin and is called the I band or isotropic band, whereas the dark band is called A or anisotropic band and contain myosin. These are the two different types of protein. Here we can actually see the segment of a muscle fiber with a nucleus present here. We can observe the sarcolemma. There's a plasma membrane of a muscle fiber. We can observe the myofibrils and definitely the dark and light bands alternately. The light band or isotropic and dark band or anisotropic band. The actin and myosin proteins are arranged as rod-like structures, parallel to each other and also to the longitudinal axis of the myofibrils. So definitely the actin and myosin proteins are arranged as rod-like structure in a muscle fiber, and definitely these are parallel to each other and also to the longitudinal axis of the myofibril. That's the unit of muscle. So here we have got a question, that is, which one of the following pairs of chemical substances is correctly categorized? Let's answer it. Calcitonin and thymosin, they are the thyroid hormones. Pepsin and prolactin, these are the two digestive enzymes secreted in stomach. Troponin and myosin, which are the complex protein in striated muscle. And definitely secretin and rhodopsin, which are the polypeptide hormones. Yes, you must have come up with the right answer, and that's just based on what we have discussed so far. And yes, it is troponin and myosin. These are the complex proteins in striated muscles. So the actin filaments which we were talking about are thinner as compared to the myosin filament. So we can actually observe the thick filaments and the thin filaments which are between the consecutive thick filaments. Alright, so the thicker filaments are definitely myosin. So these two filaments are commonly known as thin and thick filaments respectively. The actins are thin and definitely the myosins are thick. So the thin one is basically actin and the thick one is myosin, present in the myofibril. We have to understand that. In the center of each I band or isotropic band, to be honest, or this is the light band, is an elastic fiber called Zline which bisects it. So here we have the Zline that is actually bisecting the center of each I band or light band. The thin filaments are firmly attached to Zline. So that is how we can observe the structure of Zline when we observe the ultra microscopic detail of a myofibril. The Zline is bisecting the center of each I band or isotropic band or light band. The thick filaments in the A band are also held together in the middle of this band by a thin fibrous structure that is a membranous structure known as Mline. So here we can find Mline. The thick filaments in the A band, A means definitely anisotropic or dark band are held together in the middle. Okay, and that is definitely by Mline. So Z line are going to bisect the I band and Mline are going to bisect the A bands. So thin bands bisected by Z line and thick bands bisected by Mline. And definitely that's inside a myofibril, where we find thick and thin filaments. Thin filaments by actin and thick filaments by myosin. The A and I bands are arranged alternately throughout the length of the myofibrils, which we can find here. So you have the A band, lateral sides we have I bands, there's Z line at the middle of the I band, and there are thin filaments and thick filaments. Between the thick filaments, definitely we are supposed to have the M line, which we have just discussed little earlier here, the M line between the thick filaments. The portion of the myofibril between two successive Z lines. So two consecutive Z line means two consecutive lines which are actually bisecting the thin filaments. That portion is considered as the functional unit of contraction and is called a sarcomere. So here we can observe the sarcomere. So these two are the thin filaments, number 1 and number 2, and here we are supposed to have the Z line. Here we are supposed to have another Z line. So the region between two consecutive Z lines is called sarcomere, and that is definitely the functional unit of contraction of the skeletal muscles.